Okay, so welcome back from the break. Let's take a look at these two questions. They're not difficult. And then we'll move on. So what two apparently incompatible ideas have historically been used to explain embryological development? Well, those were the ideas that I introduced you to that were, I referred to as potentially diametrically opposed, that dominated the thoughts of the 18th and early 19th century. That would be preformation, the idea that within reproductive cells, there were tiny little mature miniature human beings, the so-called homunculus. The alternative was Aristotle's idea of starting with a blank slate, that the egg cell was formless, contained no pre-existing information about the ultimate shape of the animal's body as a product of embryological development. And that was known as, now I've gone blank. I'm going to have to go back to the slide. I can't. Preformation and, no, let's go back and take a look. Epigenesis. And I think I forgot to actually mention that that was the name of the idea. Now that I think about it, that's probably why I was drawing a blank. So epigenesis is the idea that we start with a blank slate or a formless idea, and through a series of graded developmental stages, you create a more and more complicated and more well-developed embryo, a series of steps. So these are the two ideas that were used historically to explain embryological development. Listan briefly described the 4 stages of embryological development. So the first stage is fertilization, when sperm meets egg, and there's the transfer of the sperm nucleus to the egg cell. Then once that fertilization stages have finished, we enter cleavage, where the resulting zygote now starts to divide. So the zygote divides into 2 cells. Those 2 cells divide again. You get 4 cells. The 4 cells divide. You get 8 cells, and so on. You pass through the solid ball of cells known as the morula, and eventually you develop a hollow ball of cells known as the blastula. From there, we move to stage 3, gastrulation, where there is a complete reorganization of the embryo and the creation of the 3 germ layers from which all the organ systems of the animal will ultimately emerge. So once gastrulation is complete and you have the 3 germ layers laid down, then you begin organogenesis, which is the development of the organ systems. So those are the 4 stages of embryological development: fertilization, cleavage, gastrulation, and organogenesis. So in the next section, we're going to talk about the first 2. So section number 2 is going to talk about fertilization and cleavage. Section number 3 will cover gastrulation and organogenesis. Then the 4th section of the presentation will talk about those 2 steps that I hinted at earlier: cell determination and cell differentiation, and what goes on to give rise to the body plan or body pattern of the final mature adult organism. So let's take a look at fertilization. So the idea of fertilization is simply to bring together the 2 haploid nuclei derived from each of the parents to create a diploid nucleus that becomes the diploid nucleus of the zygote, and subsequently through mitosis becomes the diploid nucleus found in every cell of the mature adult organism. So we need to bring together the sperm and the egg in such a way that, number 1, the correct sperm and the correct egg fuse, and number 2, the haploid nucleus of the sperm enters the egg cell. They get an opportunity to fuse, and at the same time, we trigger changes within the now fertilized egg cell that begins the process of cleavage that triggers cleavage ultimately, so that the cell begins to divide and we start to produce cells that will eventually produce the embryo and ultimately the fetus, if we are talking humans at least. So rather than read through these slides, which just details information that you could just as easily read in the textbook, or you could pull up the PowerPoint that's available in Canvas and read these slides, I'm going to go to the diagram because it's much easier for me to explain what's going on via the diagram than to read through the slides and then get to the diagram. So just please excuse me while I jump to the diagram, which is here, and we can walk through this. There are a couple of other text slides after this that explain a little bit more, but I'm going to work through everything that goes on here prior to the activation of the egg cell, which ultimately triggers cleavage. So the sperm cell in most aquatically based organisms has to find its way to the single cell that represents the egg. And that egg cell is surrounded by a layer of material that's referred to as the jellicoat. This is a protective material that prevents the egg from drying out. It also represents a barrier to the sperm cells. So one of the first events to occur during the fertilization process is that the sperm cell needs to be able to digest its way or digest away this jellicoat material to give it access to the egg cell membrane. So what we're looking at here is an exemplar of model organisms. This could be the sea urchin, it could be a sea star, it could be a frog. It's basically fairly consistent idea across the board in any organism where fertilization is external and takes place in water. So you'll notice that there is a specialized vacuole or vesicle located at the end of the sperm. This is called the acrosome, and it contains what are loosely referred to as acrosomal enzymes, but could be more correctly referred to as hydrolytic enzymes. These are digestive enzymes. When the sperm membrane comes in contact with the jellicoat, that triggers the rupture of this vesicle and the release of these enzymes, and they begin digesting away at the jellicoat. At the same time, microfilaments, I've got to remember which is the right name. Microfilaments, which are formed out of actin, start to elongate. Now, the real reason why it's important that these be microfilaments, these be actin filaments, is that if you recall from biology 1, the role of actin filaments, these are contractile, they shrink, and it is going to be the contraction of these actin filaments that are going to pull the sperm cell down to the surface of the egg cell once the jellicoat has been digested away by the hydrolytic enzymes. This all constitutes what is known as the acrosomal reaction. So the release of the acrosomal enzymes, these hydrolytic enzymes, the development or the elongation of these actin filaments to form this extension of the sperm cell called the acrosomal process, the contraction of the actin filaments to pull the sperm cell down to the egg cell membrane. This all constitutes part of the acrosomal reaction. So once the jellicoat has been digested away, the next is for the egg cell and the sperm cell to be identified as being compatible with each other. And so on the acrosomal process, this extension of the sperm cell, there are surface proteins that need to match with a series of receptor proteins on the surface of the egg cell membrane. These two sets of proteins are referred to as egg sperm identity proteins or compatibility proteins. And so it's really important in an aquatic situation, again, where fertilization is external and where the gametes, the eggs and the sperm, are simply released into the water column, that there be a way for the sperm and the egg to establish that they are from the same species. And so these compatibility proteins, the set on the surface of the sperm and the set on the surface of the egg cell, establish that compatibility. So once the sperm proteins bind to the appropriate receptor proteins on the egg cell, that triggers the actin filaments to contract and pulls the sperm cell down to the surface of the egg cell. That contact then between the sperm cell membrane and the egg cell membrane or the fusion of the membrane. So it could be just the contact of the sperm with the sperm membrane with the egg cell membrane, or the actual fusion of the two membranes, depends from species to species, triggers a depolarization event. Sodium ions from outside of the egg cell flood through channel proteins into the egg cell and collapse the membrane potential, that charge difference across the egg cell membrane. This is the depolarization event. Immediately that the membrane becomes depolarized, however, the sodium-potassium pumps in the egg cell membrane start pumping the sodium back out of the egg cell and re-establishing the polarization of the membrane. This depolarization is referred to as the slow block to polyspermy. So polyspermy simply refers to the possibility of multiple sperm inserting their nuclei into the egg cell. Obviously, if multiple sperm nuclei enter the egg cell, that resulting cell will be no longer viable. So we want to block the possibility of multiple sperm inserting their nuclei into the egg cell. So once the first sperm comes in contact with the egg cell membrane, this depolarization prevents any other sperm from binding to the egg cell membrane. It prevents any further sperm egg cell membrane fusion. But it's only going to last for about 1 minute because the sodium-potassium pumps work very efficiently to repolarize the membrane. So we need a second way of blocking polyspermy that is more permanent. And that's referred to as the cortical reaction. So this is cortical reaction. And this is referred to as the slow block. So the fast block is called fast block because essentially the collapse of the charge gradient across the membrane is instantaneous with the fusion of the two cells. So that's why it's referred to as the slow block. The cortical reaction takes place during this 1 minute window that exists while the membrane is being repolarized again. So you'll notice in the diagram that underneath the membrane in a region referred to as the cortex are these vesicles or granules, as they're commonly referred to. And again, these contain enzymes, but they also contain some soluble materials that we refer to as osmoticum. These are materials that will increase the tonicity of the environment that they're released into, and that in turn draws water in. And this is going to be an important part of the cortical reaction, this slow block to polyspermy. So if you look again at this diagram, you notice that there's this purple layer that sits immediately outside of the egg cell membrane and it's penetrated through by the receptors that are going to bind to the sperm cell compatibility proteins. That purple layer represents another structural feature called the perivitalin, or the vitalin layer. And it is held in place by these sperm cell receptor proteins. The digestive enzymes in the cortical granule will actually cut those receptor proteins so that they are no longer anchored into the egg cell membrane. That releases the vitalin layer and the jelly coat that sits above it from being anchored to the cell membrane. At the same time, the osmoticum in the cortical granules floods into the space now opened up as the vitalin layer and the jelly coat move away from the cell membrane, and that attracts water into that space, known as the perivitalin space, and that water floats the vitalin layer and what is now being converted into something called the fertilization envelope away from the surface of the egg cell membrane. So what constitutes the fertilization envelope? It is in fact a chemically modified jelly coat. It's been hardened, it's been made undigestible to acrosomal enzymes. And this is again part of the role of the enzymes in the cortical granule. They chemically modify the jelly coat so that it can no longer be digested by the acrosomal enzymes. This prevents any more sperm from attempting to digest their way through the jelly coat. And by floating this fertilization envelope away from the egg cell membrane, you further prevent the possibility of any sperm coming in contact with the egg cell membrane. So you provide a hardened barrier against further sperm penetration to the egg cell. And this all takes place during that 1 minute window that the polarized cell membrane is blocking polyspermy. But remember again, during this 1 minute, the membrane is being repolarized. So you need to carry out this cortical reaction pretty quickly. So it's a combination of the slow block of polyspermy, the instantaneous depolarization of the egg cell membrane upon contact of the sperm cell with the egg cell and membrane to membrane, and then the cortical reaction, which takes a little bit longer, but creates a more permanent barrier to sperm that prevents multiple fertilization events, blocks polyspermy. This all takes place within that first minute post-contact of the sperm with the jelly coat. Now, one of the things that we have now realized about the cortical reaction is that it is triggered by the release of calcium ions from the smooth endoplasmic reticulum. So the smooth ER is the source of the calcium ion, because it acts as a storage site for calcium ions. And we actually know that calcium ions are activators of a whole lot of other processes within cells, not just within the zygote. So it's not surprising that calcium plays a central role here in activating the cortical reaction, and as you'll see, also in activating the egg cell itself to prime it for cell division. And we can demonstrate that it really is calcium because we can trace the release of the calcium in the fertilized egg cell. So here you see an egg cell under the light microscope. You can see all the flagella of the sperm trying to penetrate the egg cell. One sperm has been fortunate enough to do so. We're about 10 seconds after fertilization. So the fast block is in place and the slow block is beginning. And we see the evidence of the slow block by the appearance of the fertilization envelope, that floating away of the vitalin and the modified jelly coat in the location where the sperm cell penetrated. So we know that this is the region where the sperm cell that managed to penetrate the egg cell actually penetrated. And you can see over time that the the development of the fertilization envelope follows a symmetric pathway going in both directions around the egg cell. And when it meets up on the other side, you have a completely formed fertilization envelope, completely surrounding the now fertilized egg cell. If we then use a system called the calcium calmodulin system, calmodulin is a fluorescent dye that fluoresces when it binds calcium. So if we inject that fluorescent dye into the egg cell prior to fertilization and then allow the sperm to fertilize the egg cell, we can see under fluorescence the release of the calcium and its binding to calmodulin, and that tells us that this is where the calcium is being released. And we see this wave of calcium spreading across the egg cell, and that parallels the development of the fertilization envelope as it moves in both directions around the egg cell. So that hints more directly at the role of calcium in triggering this process. Now, there are a number of other ways that we can determine that it really is the calcium that does this. Because if you take an unfertilized egg and inject it with calcium, so I'll put this in here. So if you inject an unfertilized egg with calcium, you see the same pattern. And what is even more remarkable is that if you anucleate the cell, which is relatively easy to do, you can use a very, very fine drawn glass needle and a micro manipulator to remove the nucleus from an egg cell. If you then go ahead and inject the egg cell with calcium, you still see the same effect. So that tells you that this is strictly a product of the release of calcium. It's got nothing to do with activation of the nucleus at all. It's not a product of any molecules that may be present in the cell. This is strictly the release of calcium that is activating the enzymes that are necessary to create the fertilization envelope and to ultimately activate the egg cell and begin the priming the egg cell for cell division. And in fact, in some of the early cloning experiments, particularly the work of John Gurdon in cloning frogs, this was one of the ways that they would prime the egg cells that they were going to insert a diploid nucleus from a donor cell into to prime it for cell division. You would simply inject some calcium in, that would prime the egg cell. You've previously anucleated it. You can then inject in the diploid nucleus. The cell is now primed to divide, and the diploid nucleus replaces the nucleus that would have developed from the sperm nucleus and the egg cell nucleus. This was this is called nuclear replacement cloning. And it, again, reinforces the fact that the calcium is what is activating the cell for cell division. So what takes place when the egg is activated? Well, basically what it does is alter the internal pH of the egg cell. It also triggers the beginning of transcription in the nucleus. Transcription and translation, transcription in the nucleus, translation in the cytoplasm. It also encourages or aids in the fusion of the two haploid nuclei. And it basically starts priming the now fertilized egg cell for beginning the cleavage process. So we can look at a timeline and see what takes place. And this timeline is based on a sea urchin or sea star. So this is the timeline based on sea urchin or sea star. By the way, if any of you are planning to get a biology degree and you are planning to go to medical school, one of the classes that you will take at your four-year school is an embryology class. In fact, most biology students are required to take it. And in that embryology class, you're actually going to create your own zygotes using either sea stars or sea urchins. And the reason why is because of this timeline, because you can do all of this in a petri dish using an artificial seawater. These artificial seawaters have been created for people who want to keep saltwater fish tanks. And we use the same sort of salt solutions to mimic the ocean for these sorts of embryological experiments. And so you will be mixing sea urchin or sea star sperm with sea urchin or sea star eggs, depending on which organisms your university works with. And you will be tracking these early embryological developments because they take place very, very quickly. So you see that the first round of cell division is going to occur approximately an hour and a half after you mix egg with sperm. So it happens really, really quickly. In mammals and in humans, it takes a little bit longer. It could be 24 to 36 hours before the first cell division takes place in the zygote. But here, it takes place a lot more quickly, and you reach the larval stage. You play out the early stages of embryological development through to organogenesis much more rapidly, something that you can actually observe in a matter of days rather than a matter of weeks or months. And so these are really good model organisms for that. And if you take an embryology class, which most of you will likely have to do if you're getting a biology degree, you'll get to make your own embryos, and you will have to spend some time both in class and outside of class watching these embryos develop over a period of days. So how does fertilization in mammals, if this is all based on model organisms that do external fertilization, what differences do we find in organisms that practice internal fertilization, like terrestrial tetrapods, like mammals? Well, most terrestrial tetrapods, amphibians, are somewhat unique in that they don't, which is the reason why we use frogs for vertebrates and representatives of terrestrial tetrapods. But in mammalian fertilization, the first thing that we need to recognize is that the egg cell is not released as a naked egg cell. It is part of a structure called a follicle. And so it's surrounded by multiple layers of supporting cells called follicular cells. We also find that there is no jelly coat, but there is a functional equivalent called the zona pellucida. And so when we have the cortical reaction and the acrosomal reaction, there it all relates to the zona pellucida, not to the jelly coat. So if we go to the diagram, here you can see the follicle cells that are supporting cells to the egg cell. The egg cell is a large cell in the middle of this cellular mass. Here you can see the sperm cell. Again, it has an acrosome. The contact of the sperm cell with the zona pellucida this time is what triggers the acrosomal reaction. The acrosomal enzymes then digest their way through the zona pellucida. You get an acrosomal process developing, that elongated structure formed by the extending actin filaments or microfilaments. They draw the sperm cell down to the surface of the egg cell. However, there are no sperm-egg compatibility proteins involved here. And the reason why is because specificity of sperm and egg is determined by choice of mating partners. So there's no requirement, given that fertilization is internal, for there to be any form of egg-sperm recognition. That didn't stop researchers early on from looking for potential proteins that act as egg-sperm compatibility proteins. There are a number of proteins that reside in the zona pellucida. They are referred to as ZP proteins. And one of the proteins, known as ZP4, was for a while suspected of being a sperm-egg compatibility protein, but we now realize that it is not, and that there are in fact no functional sperm-egg compatibility proteins. They are not required. The fast block to polyspermy is still the same. It is a depolarization of the egg cell membrane that takes place once egg and sperm membranes come in contact. We also have a release of calcium. We have a cortical reaction. The cortical granules emerge or merge with the cell membrane, release their contents. But because we don't have a vitalin layer or receptor proteins, these cortical enzymes simply modify the zona pellucida to harden it to digestion by acrosomal enzymes. There is one other feature that is unique in mammals in terms of what actually enters the egg cell from the sperm. So in our previous model organisms like the sea star and the sea urchin, even in frogs, the only thing from the sperm that enters the egg cell is the nucleus. But in mammals, the nucleus enters in and the basal body enters in. And the reason why the basal body enters in is because it becomes the structure from which the centrioles that are found in the centrosomes that guide the development of the spindle during mitosis and meiosis, during cell division. It is this basal body that is the ancestral structure to those centrosomes, and also provide the source of basal bodies that lie at the base of every cilium and every flagellum that the animal will produce over the course of its lifetime. So in male mammals, at least, the father contributes not just his nucleus, but he also contributes his basal body from his sperm cell to the egg cell, and that becomes the basis for the centrioles. So that's something that's a little bit different from most other animals. We don't see that happening in non-mammalian, mammals during non-mammal fertilizations. I'm trying to make sure that I state it correctly. One of the other things that is interesting to note too is that while we might expect the two haploid nuclei to fuse prior to the first round of cell division, in mammals, again, we find that the two haploid nuclei do not fuse until after the first round of cell division, the first splitting of the zygote into two cells. So what you actually have is reproduction of the nuclei as haploid nuclei. They get distributed to the two daughter cells, and then subsequently the two haploid nuclei fuse together in the two resulting cells, which are known as blastomeres. So that's also something that's uniquely found amongst mammals, something different. So those are the differences that we know occur during fertilization in mammals compared to our model organisms. So following fertilization, once the egg cell has been activated, once the diploid nucleus is formed in most animals, we now enter the cleavage phase. And this is a different type of cell division because, yes, you're going to see the mitotic phase, and you'll see the S phase that is associated with interphase, but you're not going to see a G1 and a G2. And so what that means is that every time the cell divides, you're not going to see any growth, you're not going to see any enlargement. What you will see is copying of the chromosomes and then division of the cell. So what really happens here is that cleavage, in the absence of growth of the two growth phases of interphase, the G1 and G2, the the net result of cleavage is to produce progressively smaller and smaller daughter cells, the blastomeres, by subdividing the existing cytoplasm of the egg cell into smaller and smaller compartments, the resulting blastomeres. The end, of course, is the blastula with its hollow space, the blastocele. But along the way, remember, we go from zygote through morula to blastula. So it's zygote, to morula, which is a solid ball of cells, to blastula. And that one, of course, is hollow. So we can illustrate this by following a cell in the process of cleavage using a light microscope and taking a series of photographs documenting the early stages of cleavage and following it through to the development of the blastula. So here are the photographs. So here is the fertilized egg. You can see the nucleus in the center. Here's the fertilization envelope around the outside. So once we go through the first two rounds of cleavage, you can see each cell has a nucleus. They are occupying the same volume of space. There's the fertilization envelope, but each of the resulting daughter cells is smaller. We are starting to subdivide the cytoplasm. When we go to the early blastula, so we've got the hollow space, you can still see evidence of the fertilization envelope around the outside there. You can see that there are many more blastomeres, but they are substantially smaller. Again, we are subdividing the existing cytoplasm into smaller and smaller cellular compartments. At the end of cleavage, when you reach the final blastular stage, again, you've got many, many small blastomeres, a large hollow space, but it's still encapsulated, very difficult to see now, but it's still encapsulated within the fertilization envelope, which will ultimately break down. But again, the whole point here of understanding cleavage is that you are subdividing the existing cytoplasm of the cell into smaller and smaller compartments, the blastomeres that are being produced as a consequence of cleavage. So one of the interesting things about the cleavage process is that in many organisms, the requirement for proper embryo, well, actually, I'll back up on that and say in all animals, there is a requirement for establishing a polarity in the organism. So there are two types of symmetry that are recognized in animals. There is radial symmetry, as exemplified by jellyfish, sea anemones, and the like. And then there is bilateral symmetry, which is the sort of symmetry that we show. If you draw a line down the center of your face all the way down the center of your body, bisecting it, that's a line down what is known as your anterior to posterior axis. What you find is that your left side and your right side are mirror images of each other. And that is the hallmark of bilateral symmetry. It is a more complicated form of symmetry because it has three axes associated with it. So you do have the anterior to posterior axis. You have the dorsal to ventral axis, which is back to front. Dorsal is the back, ventral is the front. And then you have left to right. In order to establish those three axes, which then provide positional information for the development of the body plan, the final form of the animal, you need to establish two of those axes. Because once you've got two of them, the third one is automatically fixed. So when those axes develop is variable according to the type of animal that you're working with. Different animals develop those axes, or different types of animals develop those axes at different points along the way. If you have an egg that has yolk in it, whether it be a small amount of yolk or a more substantial amount of yolk, the presence of that yolk establishes what is known as the animal-vegetal polarity. So the yolk is generally offset into what is known as the vegetal hemisphere, one half of the egg cell. The half of the egg cell that does not contain the yolk is known as the animal hemisphere and contains the animal pole. The vegetal pole is found in the vegetal hemisphere. Now, the anterior-posterior axis is not defined by the vegetal pole to animal pole axis, or so, I should say it the other way around, animal pole to vegetal pole axis. But and it's a little bit difficult to sort of describe this, but the anterior to posterior axis follows the animal hemisphere to vegetal hemisphere orientation. So the anterior end of the embryo and the final animal will be oriented in the animal hemisphere. And likewise, the posterior will be oriented in the vegetal hemisphere, but not necessarily the two poles represent the anterior to posterior axis, the line between those two poles. So the presence of yolk establishes one of the three axes that need to be established. If we look at a frog or an amphibian in general, so we could say frog, but we can say amphibian in general. So this could be salamanders as well. Salamanders have been used as model organisms as well. Here you can see the concept of the animal hemisphere and the vegetal hemisphere. And a frog egg contains what is referred to as an intermediate amount of yolk. So it contains some yolk, but it's not packed full of yolk, as you'll see it present in some other egg cells. We'll get to that. So we have the animal hemisphere and the vegetal hemisphere, and the animal pole is located in the center of the animal hemisphere. The vegetal pole is located in the center of the vegetal hemisphere. When cleavage begins, the first plane of cleavage is a vertical cleavage from the animal hemisphere to the vegetal hemisphere. And it also bisects a structural feature known as the gray crescent, which in frogs and salamanders forms on the opposite side of the egg cell to, in which the sperm cell penetrates. So the sperm cell in this case, here you can see the gray crescent. So the sperm cell penetrated the egg cell 180 degrees opposite. And that triggered a movement of a layer of melanin-containing vesicles immediately in underneath the cell membrane, and they rotated backwards towards the point of sperm contact with the egg cell, or sperm penetration into the egg cell. And that exposed a lighter area that is referred to as the gray crescent. And the appearance of the gray crescent establishes the dorsal ventral axis. So in a frog, an amphibian in general, the anterior posterior axis is oriented towards the animal vegetal hemisphere alignment. The dorsal ventral axis is more randomly produced by the random point at which the sperm enters the egg cell. That causes the melanin layer to rotate backwards, that dark cortical layer to rotate backwards, exposing this gray region, the gray crescent, and that establishes the dorsal side. And we'll see the importance of that a little bit later. The second round of cleavage also is vertical and is at right angles to the first round of cleavage. So it is, again, from animal pole towards the vegetal pole. The third round of cleavage is horizontal. And in the case of the frog, at least, because of the presence of the yolk offsets the cleavage plane into the animal hemisphere, so that that first horizontal plane of cleavage creates two layers of cells, a smaller layer of cells in the animal hemisphere and a larger layer of cells in the vegetal hemisphere. And this is strictly a product of the presence of the yolk. The yolk has a distinct inhibitory effect on cleavage. Now, in the frog, it doesn't prevent cleavage in the vegetal hemisphere. You still get full cleavage through the vegetal hemisphere, but in other egg cells where there is even more yolk material, you may not get complete cleavage. In fact, you may not get any cleavage in the yolk-containing material. I'll talk about that in a moment. But notice the effect of this offset. The resulting blastomeres in the region that was the animal hemisphere are very small and quite numerous, whereas the blastomeres in what was the vegetal hemisphere are larger and not as numerous. And the blastocele is also offset into the animal hemisphere. This is a direct product of the presence of the yolk in the region of the vegetal hemisphere. So what is the effect of yolk? Well, if you get complete division of the egg, as we saw in the frog, that's referred to as holoblastic, complete cleavage. And so even if there is a moderate amount of yolk, as we see in the frog, or just a little amount of yolk, as you might see in a sea urchin, the presence of that yolk, while it has a somewhat inhibitory effect, it slows down cleavage. It doesn't prevent the cleavage plane from completely bisecting the egg. However, in those eggs that contain a very large amount of yolk, which is basically amniote eggs of reptiles and birds, that the presence of that large amount of yolk completely prevents cleavage in the egg. So how does that relate here? I'm going to move the text box up here so I can write. So let me put this up. So in chick eggs, the yolk, what we refer to as the yolk, is actually the egg cell. So it's a very, very large cell, completely filled with yolk material. So on the surface, there's a small disc of cytoplasm, and this is where the cell division occurs. Let's say cleavage, because that's what it is. So during the cleavage stage of chick or bird and reptile embryological development, you're not going to see the yolk, the egg cell, actually being cleaved. All the cleavage is going to take place in this little disc of cytoplasm, which is no, blastodisc is the end result of cleavage there. So you don't have a formal thing called a blastula, as we would recognize it based on sea urchins and sea stars and the like. But you do have a functional equivalent that's referred to as a blastodisc, as you'll see when we get there. So let's take another break. And again, I will break the recording. So if you want to step away for a moment and clear your head, take a break, feel free to do so. When we come back from the break, I'll answer these two concept check questions. Briefly describe how the fertilization envelope develops in sea urchins. What function does it perform? And number two, what would happen to an unfertilized sea urchin egg if you injected it with calcium ions? See you on the other side of the break with the answers.